Good afternoon and boa tarde. As director of the Brazil Center at Lilas, it's my pleasure to welcome our distinguished guest today, Dr. Charles Clement, to discuss the origins of agrobiodiversity in indigenous Amazonia. Dr. Clement is a senior researcher at INPA, the National Research Institute for Amazonia. He holds a bachelor's in biology from the University of Connecticut, a master's in biology from the University of Costa Rica, and a PhD in horticulture from the University of Hawaii. After 25 years working with fruit tree improvement, he focused his research agenda on historical ecology, including plant and landscape domestication. He's published extensively on the historical ecology of Amazonian landscapes and the use and conservation of Amazonian fruit genetic resources. In fact, he's published more than 165 articles in referee journals across the globe, over 100 book chapters, and is the author or co-author of 11 books. Among the numerous honors he has received for his prodigious scholarship is the Ordem Nacional de Mérito Científico Clássico Comendador, awarded by President Fernando Henrique Cardoso in 2002. I became acquainted with Charles's work in conducting my research on the history of Guarana, and his publications on Guarana's domestication, incorporating methods of historical ecology, archaeology, plant genetics, and ethnohistory are illuminating. He's also been an incredibly supportive colleague, sharing generously of his time and his knowledge. Today's talk comes on the heels of another presentation co-sponsored by the Brazil Center last week by Justice Antônio Herman Benjamin of the Superior Tribunal de Justiça on the role of the law in protecting tropical forests. So this sequence of talks is a fitting lead up to the commemoration of Earth Day on Friday, as greater understanding of the historical evolution and the future of Amazonian ecosystems is a matter of paramount importance. I invite the audience to post questions to Dr. Clement through Facebook, and we will share them at the end of his talk. But now it's my honor to welcome Dr. Charles Clement.
Oh, hello, Seth, and everybody who's accompanying this talk. I'd first like to thank Seth very much for the invitation to be here today. It's always a pleasure to talk about plant domestication for a diverse audience. So today, I'm going to try to focus on how plant geneticists and archaeologists gather data from various disciplines to identify the origin of a given crop. And this methodology with 6 mentioned disciplines was pioneered by Alphonse de Candolle, the famous Swiss scientist who is considered the father of biogeography. And genetics was added by Nikolai Vavilov, the famous Russian plant breeder. And so we need botany in order to identify our species correctly and related species. We use genetics to study patterns of diversity. And Vavilov has always suggested that there is more genetic diversity near the origin of domestication. We use archaeology because it can give us concrete evidence of the past. This is something that genetic doesn't give us. Genetics allows inferences, but archaeology gives us concrete evidence. Biogeography helps us organize this and look for patterns in the distribution. History sometimes helps. In China, for example, with its very long written history, there's a lot of information that can be gleaned from history. In the Amazon, history started with European conquest, so there's much less information from history. And then also linguistics. When we don't have genetics or archaeology, linguistics can help, because if we associate current names, we can also try to look into the past. And so we have number of hypotheses about where different crops may have originated in lowland South America. And most of these are in what we call Greater Amazonia. And if you look at this map, you see a lot of the names, which may be unfamiliar to you, but they also have question marks after the name. And that means we're not quite sure. Where there is a name with no question mark, that means we've identified the origin of domestication. And what's interesting about this map is that most of the crops are located around the periphery of the Amazon. And only Guarana, which is the topic of Seth's new book that's coming out, was domesticated in the center of the Amazon. Okay, and so looking at this list of hypotheses and where human populations were located at the time of European conquest, we can associate origins with dispersals. And this is because human populations that grew quite large in the Amazon by European conquest accumulated genetic resources or agrobiodiversity in their villages. And when there was a lot of humans and a lot of agrobiodiversity, we can identify centers of diversity. And when there's a little bit less, we call them regions. And when the geographic extent is very small, we call them microcenters or minor centers. And this is terminology that comes from Vavilov and other students of crop origins. And so archaeology, as I said, can give us concrete evidence, but sometimes it's not in Amazonia. Botanical archaeology is very new here in the Amazon, and we still have very little information. But the Pacific coast of Peru and also the mountains, the Andes of Peru, have been well studied by archaeologists for nearly 100 years. And what they found on the Pacific coast is that the Murupi hot pepper arrived on the Pacific coast about 10,000 years ago, and manioc and sweet potato shortly thereafter, and guava about 8,000 years ago. And what's important about this is that none of these are native on the Pacific coast. So what happened is the domestication started earlier in Amazonia, and domesticated varieties were taken from Amazonia to the Pacific coast of Peru, where they were grown in irrigated production systems. Okay, so archaeology, even from the Pacific coast, can tell us about Amazonia. And the new work in the Amazon has started to identify different crops from the early Holocene. And lereng is a root crop, which is almost always identified very early in South American archaeological sites in the lowlands. Piquia is a major, large-statured fruit tree, similar in stature to the Brazil nut, which is the iconic nut tree of the Amazon, and guava also. So we're talking about the early Holocene is between 7,000 and 10,000 years ago in Amazonia. We find some of the things that were expected from what we know about the Pacific coast. And interestingly, in their middle Holocene, rice was starting to be domesticated. And when we talk about rice at the world scale, it's well known that rice was domesticated in Southeast Asia and distributed up into China and then into India. Rice was also domesticated in Africa, in Western Africa. And so this new finding from 2017, puts South America into the world rice map. And then manioc, of course, which is the major energy crop, and squash, which is always an early domesticate, are also appear in archaeological sites here in Amazonia. So as I said, these are still very recent and not very abundant research, but it's coming on very nicely. The botanical archaeologists are doing a very nice job here in Amazonia. So let's look at how we can identify the origin of a crop. So this is manioc. It's a primary energy staple crop of the Amazon. And the yellow, gritty stuff in the hand of the person showing you this manioc flower is how it's consumed here in the Amazon. So the roots are transformed into this flower, and archaeologists can find evidence of manioc by the tools that are used to process the root into the flower. But geneticists and biologists can use other information. So this is a very famous article published at the turn of the millennium by Ken Olson, who is now at the University of St. Louis in Missouri, and Barbara Schall, who I think is in California, I don't remember. But the model that they used for this study is classic. What they did is they collected the wild relatives of manioc across southern Amazonia, where it grows as a vine in open forests at the edge of the savannas, and sometimes in the savannas themselves. And then they sequenced the gene, and they used 5 microsatellites, which are small pieces of DNA that are easily multiplied in the laboratory. And they compared the wild populations with a nuclear collection of domesticated manioc. And they found that all of the domesticated manioc has the same enzyme sequence and the same microsatellites as these black squares in the map. And so they hypothesized that manioc started to be domesticated in what is today Rondônia state and Mato Grosso state, and perhaps a little bit of Acre state in southwestern Amazonia. And you might wonder about Bolivia. And Bolivia is certainly involved, but was not involved in the collection expeditions. Okay, so this is a classic study for determining the origins of domestication of any crop. And this kind of design has been used by many groups around the world, including our group here in Brazil. When manioc was domesticated, 2 varieties developed. One is called sweet manioc, and the other is called bitter manioc. And this has to do with the amount of hydrogen cyanide that's in the root. And hydrogen cyanide is very toxic, but it's also bitter. So you can chew on a little piece of manioc, just a little bit so you don't kill yourself. And you will notice that in the bitter varieties, you immediately notice that it's bitter. But in the sweet varieties, you have difficulty telling whether there is any bitter substance. Huh? And so you can look at this map, and the sweet manioc are the green bars, and the bitter manioc are the orange bars. And so we have information here about manioc in the year 2000. Different kinds distributed in different parts of the Amazon. And if we look at the linguistics, we can get an idea of who thought that manioc was important at different times. So the larger the circle, the red circle, the earlier that people thought manioc was important. And in this figure in the lowland Amazon, there are two groups of Arawak speakers, which the Arawak language trunk is one of the largest, most widely distributed languages in lowland South America. And so 4,000 years ago, the Arawakan people in northern Amazonia and in southwestern Amazonia, which is near where Olson and Schall defined the origin, thought that Manic was important. Okay, but we can't get all the way back to 8,000 or 10,000 years ago with linguistic data. So we can get back 4,000, maybe 5,000 years ago. And so this is important to show that different linguistic groups, different ethnic groups in the Amazon adopted manioc at different times. So manioc is not a consensus. You can see a large group of Macro J speakers in central Brazil in the savannas did not think manioc was important until recently, okay? And so manioc has different histories. So a colleague of ours, Gilda Moulin, collected about 500 some manioc varieties in Brazil, and she used microsatellites, similar to those used by Olson and Schall, to genotype all of her samples. And with a program which is based on Bayesian probabilities, she could group these different varieties into different groups of manioc. And so when she runs the program, the first group that comes out, the first two groups, are the bitter and the sweet, which we call Mandioca brava is the bitter and Macaxeira is the sweet. And these names are from Tupi. And so this graph on the top is divided in half. And on the right side is green and blue, which are the sweet maniocs. And on the left side is red and yellow, which are the bitter ones. So the first group is the division between bitter and sweet. So she asked the program again, how many groups are there? And the program said, okay, we can divide the sweet into two groups. And then at a later probability, we can divide the bitter into two groups. And so this shows that not only are the bitter and sweet dispersed differently in the lowland South America, but there are different groups of sweet and different groups of bitter. And so she put this into what we call a dendrogram and looked at the relations between these groups. And what she found is that the sweet manioc is most closely related to wild manioc. The wild manioc that I said grows like a vine rather than a bush. And all of the bitter manioc is derived from the sweet manioc. And so this is something that's very interesting because the wild manioc is extremely bitter. So what this graph, this dendrogram tells us is that manioc started to be domesticated to be sweet, and at a later stage in time, it was re-domesticated to be bitter. And a well-known archaeologist here in Amazonia, Manuel Arroyo-Callen, hypothesized that manioc is domesticated to be sweet in home gardens and grows as a minor crop everywhere in the Amazon. And if you remember the map about how it's differently distributed, in the Western Amazon, sweet manioc is more common than bitter manioc. And that same region is where maize is more important than manioc. But where manioc is more important than maize, bitter manioc dominates. Okay, so we have a very complex story of domestication and re-domestication of this major crop. And today in Brazilian Amazonia, bitter manioc is more important. And in other parts of Brazil and Peru and Colombia and Bolivia, sweet manioc is more important. So let's go on to another starchy crop. This is peach palm, which is a crop that I've worked with for about 40 years. And on the left side, you see very small fruit on a fruit bunch. And this is what wild peach palm looked like when the Indians started to domesticate it 10,000 years ago. And the larger fruit around the small fruit bunch are a medium-sized domesticated fruit. And today, these fruit on the right, photo on the right, are consumed or can be made into flour or into cakes and other different kinds of home uses. But mostly it's consumed as a cooked fruit. Okay, so we worked for a long time mapping the different kinds of peach palm in the Amazon. We mapped the wild varieties and we mapped the domesticated varieties. And across southern Amazonia, the red shaded area is the area of distribution of the little red fruit that was in the photo. And the blue area, which has a disjunct distribution, part of it is in Western Amazonia and part of it is on the other side of the Andes in Ecuador and Colombia. And this is the incipient domesticate, which was originally described by the botanists as a wild variety. And up in Venezuela and Colombia, there's another wild type, which is not involved in the domestication. And then in the Brazilian Amazon and Colombian and Ecuadorian, Peruvian Amazon, you see different areas with different shadings. The orange shading in the center are the large fruit that I showed you. And in some cases, they get to be very large. A single fruit can weigh 200 grams, and it's enough for an entire breakfast. Okay, so these different domesticated races of peach palm are distributed in different areas. And you can tell that we haven't collected the entire Amazon because there's lots of areas with no shading at all. So one of our students did the same kind of study that Jill DeMoulin did. And I've already explained how this works. And Vanessa Reyes used 17 microsatellites, a little bit more than Jill DeMoulin did with manioc. And she analyzed it the same way. And she found that the first group that separates out is Eastern Amazonia, Amazonia Oriental, the blue group on the left. The second group that separates out is the green group in Central America. And Amazonia, and especially Western Amazonia, are divided into two groups, okay? And so she put this onto a dendrogram, just like Gilda did. And she found something that suggested that peach palm had been domesticated 2 times. And this is common in crop domestication. Okay, and so her hypothesis was that the Eastern Amazonian types of peach palm with the blue coloring in the figure were domesticated in the upper Madeira River region. And the other peach palms were domesticated further west and possibly even along the Ucayali River in Peru. And then these were taken into Western Amazonia and finally across the Andes into Central America. So to verify this, Michele Cristo de Araújo looked at the chloroplast. And chloroplast gives us a little bit more ancient information than the microsatellites, which are from the nucleus. And so she sequenced a chloroplast in the same samples of peach palm that Vanessa Hayes had used. And she added one additional species here, which is the little pinkish or purple dot on the right side of the figure. And this is a unrelated Bactris species. And so what she showed is that this process of domestication started in the upper Madeira River basin, as Vanessa had suggested, but that there was only one domestication. And this domestication gave rise to both the Eastern Amazonian and the Western Amazonian peach palms. And so what she showed and what we published a couple years ago is a figure that looks like this. So from the same region in southwestern Amazonia, peach palm was domesticated and then dispersed across the Amazon and finally up into Central America. So now we'll look at cocoa, because when people think about the crops of the Amazon, most of them think that the most important crop is cocoa because that is where chocolate comes from. And most everybody loves chocolate. But most of you who love chocolate have never actually had chocolate the way the Mayan Indians invented it. And that's the little cup of liquid on the right side with a nice hot pepper. So chocolate was originally developed as a drink. And Theobroma is the name that Carl Linnaeus gave to the genus when he described it in his his first species volumes back in the 1700s. And Theobroma means drink of the gods. And so this idea of chocolate as a chocolate bar, and we just came through Easter with all those chocolate eggs, right? Chocolate originally didn't have sugar in it. It had vanilla, hot pepper, and Anato coloring to give it a little bit of a red tinge. And it had maize to thicken the liquid. No sweet stuff. It was a whole different chocolate experience than you've had before. And Juan Montemayor, because chocolate is so important, Juan Montemayor was able to work with 106 microsatellites. And he went to look at how the genetic diversity of chocolate, of cocoa, is distributed in lowland South America and up into Mesoamerica. And when he started, cocoa geneticists considered that there were only two varieties of cocoa. The Criollo, which is the yellow dot in Central America and northwestern South America, and all the rest, which were called forastero types. And so what Juan Montemayor and his group showed is that the forastero types are very diverse, and that in lowland South America, there are 9 different groups of genetic diversity of cocoa. And all of these are used by people. And so if you look very closely in his data, Juan Montemayor was not worried about domestication. He was looking at how diversity is distributed. But I looked at it in terms of domestication. And what I found was, later studied by another group led by Everett Thomas, that during the process of domestication, the number of alleles in domesticated populations is reduced by human selection. And if you look in Mesoamerica, in Costa Rica and Mexico and what have you, all of the dots are very light colored because there's not very many alleles. And in Western Amazonia, you see that most of the dots are dark colored. So there's a lot of allelic richness in Western Amazonia. But as you go east down the Amazon River, you lose genetic diversity again. And so what I suggested, and then Everett Thomas showed quite clearly with this figure, is that cocoa was domesticated twice. Once when it was taken up into Mesoamerica and the Maya people invented chocolate. And once when it was taken downriver and chocolate was not invented. And cocoa is used as a fruit. It's not only used to make chocolate. So the white pulp around the seeds is very juicy and very sweet, and people just crack open a pod and suck on the seeds. And this is how it was used throughout lowland South America, including in southern Mesoamerica, until the Mayas invented chocolate. And so we can see that cocoa was also domesticated one time, and then, or started to be domesticated one time, and then dispersed along two different routes for its final domestication in Eastern Amazonia and in Mesoamerica. And this is Guarana. This is the subject of Seth's new book. And one of the things that got Seth interested is that there is a legend about the origin of Guarana. And this legend is summarized in the picture graph on the left side, where a young Indian boy is killed. And we won't go through the whole story, but his mother was a medicine woman and has magical powers. And she buried her son's eyes and also his body. And from the left eye, germinated, shall we say, a false Guarana plant. From the right eye, the real Guarana, which is a magical plant. And from the rest of the body, all of the other plants and animals that are important to a group of Tupi Indians that originated at the same time. These are the Satare Mawe indigenous people in central Amazonia. Remember that I said Guarana is the only crop that was domesticated in central Amazonia. And it was domesticated quite recently. The Satare Mawe arrived in the region that they now live in about 1,000 years ago. And they identified Guarana, and Guarana became an emblem of their identity as an indigenous group. And I said, I said that Guarana is magic. And the Satare Mawe say it's magic for a series of reasons. But I say it's magic because The real Guarana, the domesticated Guarana, is a high-level polyploid. And it has to be a magic plant to fit 210 chromosomes into a tiny little nucleus, where all the other species of the Paulinia genus only have 24 chromosomes. So this shows that the Guarana was domesticated when the Saturday Maui people identified this polyploid, okay? And it became so important to them that Guarana is a major part of their cultural identity. So there are other things in the Amazon besides fruit and crops. And one of them is a gourd, which is used to make recipients. And indigenous people will have a very fine artistic tradition of decorating gourds. And so in the floodplains of Amazonia is a tiny little gourd, which is shown on the left side. And there's a similar tiny little gourd in Mexico. So two different well-known botanists hypothesized that tree gourd was domesticated in Mexico or in Amazonia. And the Brazilian botanist chose Amazonia, and the US botanist chose Mexico. And the young lady holding a gourd in the figure in the center shows that tree gourds can become very large, okay? And so the question is, where did the domestication start? And Priscilla Morera was a student of ours who did part of her work in France with the group of Yves Vigoureau in Montpellier. And she sequenced a part of the chloroplast. And she showed that neither Mexico nor Amazonia is where the tree gourd started to be domesticated. But because she didn't have a sample of tree gourd from Nicaragua, Costa Rica, Panama, and northern Colombia, she couldn't identify the origin of domestication. But she did show that it was taken from its origin somewhere in Mesoamerica up into Mexico and down into the Amazon. And that it came into the Amazon via the Orinoco River. Okay, and so the orange area, in this figure, the darkest red area up in Mexico is one end of the distribution passing through through southern Mesoamerica into Colombia, and then down the Orinoco River into the Amazon, where it diversified and hybridized with the other Amazonian small tree gourd that I showed in the first picture. So we have a very complex story of domestication and distribution and hybridization. Okay, so this is a very interesting story of domestication of a technological crop. And another crop that's important is Anato. And Anato is widely used today as a food coloring. People are getting away from synthetic food colorings and going to natural ones. And Anato gives a very nice red, orange, yellow food coloring. And so Juliana Linz and the same Priscilla Morera collected Anato in large parts, in various areas of the Amazon, and gave the samples to Gabriel de Guglielmani, who used microsatellites to analyze wild and domesticated types of Anato. And what he found is that Anato probably started to be domesticated in Rondonia, the same region where manioc started to be domesticated and peach palm started to be domesticated. So southwestern Amazonia is a very important part of Amazonia. So this study has not yet been published in its final form, but is now in review. And Amazonia also has grapes that grow on trees. And this Amazon tree grape is especially important to an indigenous group in Western Amazonia, the Chikuna indigenous peoples. And Hermísia Pedrosa worked on this for her master's degree, and she pulled together information on the biogeography of tree grape. And this is information from the Amazon Tree Diversity Network permanent plots. And the green dots are permanent plots on upland soils, and the orange dots are in floodplain. So this is a species that is adapted to two extremely different varieties, two extremely different environments in Amazonia, the floodplain and the uplands. And the yellow dots are where tree grape is cultivated, okay? And the Chikuna indigenous group is located around the triple frontier of Colombia, Peru, and Brazil in Western Amazonia. So Hermísia studied the morphology of this domestication, and she also studied genetics with chloroplast markers. And these two large pizzas in the center of the figure are wild and domesticated tree grape from the Upper Solimões River. And so with this, she showed relatively well. It would be nice to have nuclear markers also, but the chloroplast is pretty good at showing us that the domestication started in Western Amazonia, associated with the Chikuna indigenous group. And Brazil nut, as I mentioned earlier, it's one of the iconic trees of the Amazon. It's a large emergent tree. And you've all had the Brazil nut seed. It's not actually a nut, it's a seed. And so you've had experience with it, although you may never have actually seen a tree. No? And Everett Thomas, who's the same person who worked with that study of peach palm I told you about, is very interested in modeling where you might find a given species during the last glacial maximum, before humans started distributing many species across the Amazon. And in this map, the green areas are where Brazil nut may have been most abundant during the last glacial maximum. And the dark squares, and sometimes yellow squares, are again allelic richness. And so in northeastern Amazonia and in southwestern Amazonia, the Brazil nut has the highest genetic diversity. And remember that I said that Nikolai Vavilov had suggested that this would be the origin. And one of these areas in southwestern Amazonia models very nicely to the last glacial maximum, but northeastern Amazonia doesn't. And Everett Thomas is very attached to his models, but a famous modeler from the beginnings of the last century said all models are wrong. So some of them can be interesting. And this is certainly interesting because of the ship in southwestern Amazonia. But we cannot discard northeastern Amazonia just because one model said that there is no chance of it being there during the last glacial maximum. So I've talked about a half a dozen species. The map I showed you earlier has 20 species with lots of question marks, but at least 80, perhaps 90, maybe even 100 species started to be domesticated in the Amazon before European conquest. They're not even on this map because we have no hypotheses yet about where they started to be domesticated. But we do know that they were all taken, all accumulated by different indigenous peoples before European conquest. Because the diversity of crops, different kinds of crops, different varieties of each crop is what allowed native Amazonians and native Americans in general to produce an abundance of food in their farming systems. And you remember that at this time there was no fertilizers, no chemical fertilizers, no pesticides of any kind. So diseases and pests were managed with diversity. And this is the importance of diversity to these people in these areas that are shown in this map. Okay, so this is the story I wanted to tell you. This is an ongoing story. There are people today in Amazonia and in other parts of the Americas and even in Europe studying the origins of different Amazonian and American and African and Asian crops, okay? So I thank you very much for your attention. I hope I didn't put anybody to sleep with this. And if anybody has questions, please put them on Facebook and Seth will be kind enough to transmit them to me here. Thank you very much.
Thank you, Charles, for that wonderful talk. Maybe I can start by asking you a little bit about sort of the methodology that you and your teams use. You mentioned these various disciplines that are critical for carrying out this type of multidisciplinary study. I'm curious to know, do you bring in specialists in all of those fields, or does each researcher try to acquire an expertise in the various fields? And my second question is about the use of technologies and how, and to what extent recent technologies have helped you and your teams and your students and your colleagues to carry out this type of very expansive and extensive research.
Okay, thank you, Seth, for these questions. When possible, we like to bring in specialists from different disciplines. And so our group today works closely with archaeologists and anthropologists. We have not worked very much with historians. We have worked with linguists. And so when possible, we prefer to have a specialist because amateurs often make mistakes. And while I might understand the genetics that I showed you in most of the stories, my grasp of linguistics and history and anthropology is fragile at best. So having a specialist is always the best option. And around the world, the groups who are working on domestication of various crops here and there also like to work in multidisciplinary groups. And most of these groups will combine at a minimum genetics and archaeology. And this ties into your second question about technologies. The technologies available in genetics have become remarkably sophisticated in the last 2 decades. I still remember when my major professor at Hawaii said that I could only get a PhD in genetics if I extracted enzymes to study enzymatic variation in peach pulp. Today, my students are extracting DNA and sequencing whole genomes. So the sophistication of these techniques has developed remarkably over the last couple decades. And the archaeologists have not been left behind. They have developed their own techniques. One of the most important things developed in the last 20 years is the study of phytoliths. Phytoliths are microscopic pieces of silica that almost all plants produce within their tissues because they absorb silica from the environment, but they don't have any use for it. So they store it as these small fragments of glass, okay, essentially. And in some plants, these phytoliths are extremely diagnostic. And Dolores Peperno is a famous archaeologist at the Smithsonian Institution in Washington, who is one of the pioneers. Deborah Pearsall at the University of Missouri is another pioneer. And Peperno and Pearsall have shown that in maize, in squash, and in a few other crops, you can identify varieties of maize by phytoliths. And so they have shown how maize was domesticated and domestication changed the phytoliths and how it was distributed, and different phytoliths are distributed in different parts of the distribution of maize. Well, squash is not quite as sophisticated, but archaeologists are now starting to work with geneticists also, because archaeologists and geneticists found that you can extract usable DNA from archaeological samples. So if an archaeologist finds a maize cob, for example, in an archaeological site in the coast of Peru, where it's very well preserved, DNA can be extracted and sequenced and compared with modern DNA. So this is happening in many species now, and has become a very formidable tool to combine the two disciplines of genetics and archaeology. So another new study just 2 years ago showed that if you study charcoal carefully, you can determine whether it was produced at high temperature or low temperature. And this is important because indigenous peoples used low intensity fire to manage environments so that they could produce food crops. Because they didn't have herbicides and they didn't have steel tools, what they did was they weeded intensively, or every week they'd be out there in the field weeding. And as soon as a pile of weeds dried enough, it would be burned in a low intensity fire, because you didn't want a high intensity fire because it would damage the plants growing around it. And so this distinction between low and high intensity fire is very important to interpret how people manage their landscapes in the past. Okay, so the archaeologists are developing very sophisticated technologies also. Okay.
Thank you. Paloma, I think, has a question.
Yes, and let me turn off my cell phone. Clemens, my question has to do a lot with what you were mentioning about new technologies and new tools used recently. What do you say are the biggest misconceptions that have been rectified with the use of these new technologies and tools? And the other question is, you also mentioned people from different academic training, different disciplines working there. In what areas do you think there's least consensus on the findings between anthropologists and archaeologists and geographers? And how do you resolve this lack of consensus?
A very nice second question, Paloma. But let's look at the first one, because technologies are just tools. And it doesn't matter how sophisticated your technology is if you don't ask the question appropriately. And a famous US geneticist named Richard Lewontin, who was at the University of Harvard, who wrote at the turn of the millennium that development of these genetic tools has made geneticists concentrate on questions that can be answered with these tools, and left out of the picture are questions that cannot be answered with those tools. And domestication, for example, is a process of human interaction with other species. And so if we look at how a geneticist imagines domestication, it's all about changes in allelic frequencies and changes in the size of a given part of the morphology of a plant or an animal, okay? And this is not really how people domesticate. Nobody goes out to reduce allelic diversity or increase the size of a tree gourd. Or Charles Darwin was very clear about this when he wrote about the origin of species and later on the variability of crops and animals in domestication. He said that people like diversity. They collect different kinds of tree gourds, for example. And by having those all together in one place, these can hybridize. And by chance, a hybrid might be better than what was already in the collection. And so people will select the new hybrid. But it's because they like diversity. It's not because they're looking for larger and larger tree gourds. In fact, some of the most important uses of tree gourd today are for the smaller sizes, which are used in musical instruments. Okay, so we have these wonderful sophisticated technologies which are biasing how we study domestication. And some anthropologist, Anna Singh, for example, has criticized the geneticists and archaeologists for ignoring the human dimension, because domestication is a human activity and it's carried out by anybody who has a garden and likes plants and animals. Okay, so the technologies are sophisticated, but sometimes we lose sight of what's really going on because we concentrate too much on our technologies. Okay, and this also ties in with the second question that Paloma gave, which is how to integrate social scientists and natural scientists. Well, social scientists, I'm speaking now, of course, as a natural scientist. Social scientists are strange people, but social scientists think the same about natural scientists. Natural scientists are very strange people also. And sometimes, even though we speak English or Portuguese with each other, we're not actually speaking to each other. We're speaking past each other. So a well-known anthropologist here in the Amazon, Daryl Posey, who died at the beginning of the millennium, once told me that it can take months to negotiate the meaning of words when you are working with an indigenous community. And it turns out that his comment is just as valid for a natural scientist working with social scientists or vice versa. So it's a question of how our different backgrounds and worldviews need to be negotiated so that we can converse with each other and find outcomes that are mutually satisfactory. Okay, and so working with a very multidisciplinary group, which is common in domestication research, is a challenge because of this, because we come from different backgrounds, different ways of being trained, different world views of how the academy influences our own activities and can influence the world. Okay, so it's a challenge. I'm still learning how to talk with anthropologists, and it's always illuminating when I finally get something right. Huh?
Thank you. We have a question from the audience. Nailza Steiner writes, hello, Charles. Thank you for your talk. In the studies that you show us, what is the concept of varieties?
Very good question. And Nailza Steiner is in Santa Catarina, I imagine. And so what is a variety? That's a good question. It's a question that is also asked about species. And it turns out varieties exist and species don't. And biologists took a long time to figure this out. So what is a variety? A variety is a group of plants of the same species which have morphological characteristics that show that they are more similar to each other than they are to other plants of different varieties. And if we look at this with genetic techniques, we can often find that a variety A comes from one genetic lineage, while variety B comes from another genetic lineage. And this is how phylogeneticists are considering species today. So different lineages have greater or lesser similarity and can be grouped into something that we call species. But genetically, they're lineages. They're not necessarily interbreeding, okay? Varieties, however, because they tend to be at the ends of these dendrograms that I showed you and are closer to the end of a lineage, are always interbreeding. So not only do they look morphologically more or less similar, but they cross easily and create more variability within that variety, okay? And so this very nice question from Nailza is about how we compare these different things, okay? Because in order to look at the history of domestication, we have to compare the varieties that exist today, okay?
I'd like to ask, maybe this might be the last question because we're coming to the end of our program, but I'd like to ask you, if you don't mind, since you've had such an illustrious career, to reflect on your transition into historical ecology. You said that you'd worked for many years in other aspects of research, and then you turned to this focus. So I'm curious to learn about what led to this transition. And the second question was about, so much of your work looks at the deep past of Amazonia and this long history of agroecology and agrobiodiversity. But when we think about the future of Amazonia, what is your prognosis? You know, what is your for what you see and some of the concerns and some of the things we can learn from your research?
Well, let's go to the first part because that's easy. Talking about the future is always complicated. When I started working at INPA 40 years ago, 45 years ago, I was put into a group that was starting to study agroforestry. And agroforestry are the combination of crops, some tree crops, some annual crops in a given plot. And this is how most farmers around the world have produced food and other necessities through history. Well, what's called agriculture is actually something quite new, and agroforestry is quite old. And within this group, I transitioned into the person who looked for different varieties of crops to put into our different agroforestry systems. And so I started to travel around Amazonia looking for useful in terms of the modern market. All of them were useful to the indigenous peoples and Ribeirinho communities who produce them. But the modern market is not always the same as traditional subsistence. So I visited numerous parts of the Amazon. And during these numerous visits, I discovered, and reading up on previous work, I discovered that others had found the same thing. The Western Amazon turns out to be where the majority of fruit trees will have the largest sized fruits. Okay, so size of a useful plant organ is one of the most evident things in domestication. Humans tend to like large things. And so I started to concentrate on Western Amazonia. And that's what led me into domestication studies. Why are these people in that region domesticating more tree crops? And so this led into anthropology, and then it led into archaeology. And at the same time, I was part of this group working on agroforestry and developing fruit for the market. Well, as you know, Brazilian history has been rather disturbed along the last couple decades, last 4 decades since I was there. I arrived during the military regime, and I was here when Brazil re-democratized. And when you have democratic regime, you don't always know if you're going to get a good president or not, because a presidential campaign is more about marketing of people than it is of ideas. And you recently went through the same thing in the US, where you had Trump, and we currently have Bolsonaro. But in the last 40 years, we've had good and bad presidents, and of course I count Bolsonaro as one of the worst. But this affects institutions. And so institutions will suffer or advance during these different governments. And several of the presidents over the last 40 years will cause more suffering than advances. And part of the suffering led to the collapse of the group that was working on developing fruit trees for the market. So when this happened and our experiment station became almost non-viable, other colleagues decided to leave. And I said, well, no, I can stay on, just change my focus. And so I had been interested in domestication for a long time, so that became my primary focus, rather than developing fruits in agroforestry systems for the market. Okay? And so this ties into your next question. Looking at Bolsonaro, we look at a figure who has absolutely no interest in the future of anything except his own family. He's not interested in the future of Brazil. He's definitely not interested in the future of the Amazon. And he's, if possible, he's even less interested in the future of indigenous people and local communities in the Amazon. And you all know that Amazonia is a pivotal area in climate change because it stores carbon. And if this carbon is released to the atmosphere, as it is being released continually, the global climate is warming and the flow of humidity around the planet is changing. And what is expected, even if we get a new president who suddenly decides that we're going to save the Amazon, even if we get a new president, it may be too late. Very important climate scientists are talking about a tipping point. And those of you who follow the IPCC know that the most recent report from the IPCC said we only have about 5 years to get serious. And they said that about the same time that Putin started the war in Ukraine, right? And so everybody's worried about the war now, and they're not worried about climate change. And so the likelihood of global industrial societies getting serious in time to avoid severe climate change is very small. And when the tipping point arrives in the Amazon, and it could be within the next 10 or 20 years, the Amazon will tip into savanna. And the forest that I know and that you've all read about, and you can see as a nice big green area on the map, will disappear. And a savanna type of vegetation will take its place. Ooh. For agribusiness, this might not be so bad because it will change the climate of Amazonia to be more appropriate to grow soybeans. Imagine that. Yeah? And so if global industrial societies do not get serious, the future is savanna and soybeans in what is today Amazonia. Now, some of my students think that I'm a pessimist, but I'd like to remind you that the difference between a pessimist and an optimist is that pessimists tend to have better information.
Thank you. That does bring the program to a close. I did want to thank Paloma Diaz for her assistance in organizing this event. And I wanted to thank once more Charles Clement for this interesting talk and all of the very important work that you do. And I wish you all a good afternoon or a good evening.